Good morning, everyone. Welcome to another webinar series from the School of Medicine, Faculty of Medicine, Bioscience, and Nursing. If you are watching this, please give us a like and share this video. Also, do follow our MAHSA University social media and get notified for our future events and latest webinar. So, in today's webinar will be how convenient is PCR test? Before we begin, let me introduce the speaker for today, Dr. Kindar Lee. Dr. Kim Tun currently works as a lecturer in the Department of Pathology in the Faculty of Medicine, Bioscience and Nursing at MAHSA University. She is a pathologist with a keen interest in teaching and research in pathology, and she is a recognized pathologist in Myanmar. She has been involved in teaching and research for the past 16 years. She's regularly attends and takes part in monthly clinical pathology conferences. She has published more than 10 research journals and is currently working on the impact of worm infestation in primary school children. So, without further ado, let's hear it live from Dr. Dali. Doctor?
Hello, good morning everyone. Today I'm going to present the how convenience of the polymerase to diagnose in coronavirus. Is it actually? On January 8th this year, the pathogens of severe and explained viral pneumonia was identified by sequencing as a tentatively named novel coronavirus. On February 12th, the International Committee on Taxonomy of the Virus declared that the 2019 corona was officially named SARS coronavirus 2, SARS-2. So at the same day, WHO, World Health Organization, declared the disease caused by SARS-CoV-2 was officially named as coronavirus disease 2019, COVID-19. So till now, about 4.18 million of the cases have been confirmed. 211 countries, the territories such as USA, Southeast Asia, including severe death case there. COVID-19 coronavirus real-time PCR kit is IVD reagent relying on fluorescent PCR technology and aiming at qualitative detect SARS-CoV-2. The kit contains oligonucleotides primer, double label hydrolysis probe, and quality control of the PCR reactions. So here is the virus look like this. So as results of the steady research related to the COVID-19 must be available for analysis and application, also as one of the mandatory requirements to have the approval of some ethics committee for the research. So to minimize the death toll of the COVID-19 pandemic, it's necessary to improve the quality of basic service, water, electricity, and remove the obstacles to food production, distribution, and it's increased the laboratory with the PCR-RT testing around the country. So these are the tests available to get the diagnosis for COVID-19. So we can test taken from the swab. Also, we can test taken from the blood, centrifuge after the PCR, RT-PCR. RT means a real time, and we can also test by the rapid test. Also, we can test by the ELISA. RT-PCR test to detect the severe acute respiratory syndrome, SARS-2, COVID-19 is the operational gold standard for detecting this disease. And RT-PCR assay in UK have analytical sensitivity and specificity of greater than 95%. So there is a reference captured here. So what is real-time PCR? Real-time PCR is a nuclear-derived method for detecting the presence of specific genetic material in any pathogen, including virus. Originally, the method used radioactive isotope makers to detect the target genetic material, but subsequent refining has led to the replacement of the isotopic labeling with specific markers. Most frequent fluorescent dye. So once we put the fluorescent dye, it's more obvious to check. So this technique allows scientists to see the result almost immediately, whereas the conventional RT-PCR only provide result at the end of the process. Real-time PCR is one of the most widely used laboratory methods to detect the COVID-19 virus. While many countries have used the real-time PCR for diagnosing such as Ebola virus, Zika virus, and many need support in adapting this method for the COVID-19 virus. The coronavirus, which caused COVID-19, only contain RNA, so which means that they rely on infiltrating healthy cells to multiply and survive. Once inside the cell, virus use its own genetic code. RNA, in the case of the COVID-19 virus, to take control and reprogram the cells, turning them into the virus-making factory. So in order for a virus like COVID-19 virus to be detected early in the body, using as a real-time PCR, scientists need to convert their RNA to DNA and reverse transcriptions. So because the DNA have a copy and we can do the amplifications, amplify, which is the key part of the real-time RT-PCR process for detecting the virus. Why use real-time PCR is a highly sensitive, specific, reliable, right time, and also cheap. Compared to other available virus isolation method, real-time RT-PCR is significantly faster, has a lower potential for contaminations or error, as the entire process can be carried out within a closed tube. It contains to be the most accurate method available for the detection of the COVID-19 virus. What PCR and how is different from real-time? RT-PCR is variation from the PCR. The polymerase chain reaction, the two techniques are used in the same process, except that RT-PCR has an added step of the reverse transcription of the RNA to DNA. RT is the real-time. Reverse transcription also the RT, it means denotes a reverse transcription to allow for amplification. This means PCR is used for pathogen that already contain in the DNA for amplification, while RT-PCR is used for those containing RNA that needs to be transcribed to DNA for amplification. Both techniques can be performed in real time, which means results are visible almost immediately. Conventionally, results are visible at the end of the reaction. So PCR is one of the most widely used diagnostic tests for detecting viruses like Zika virus, Ebola virus. So since the COVID-19 virus only contain RNA, so real-time PCR is the very convenient to use. So research has already published. So let us go back to the basic first. So as we all know, all living organisms are made of cells. So each cells has 20 pairs, 23 pairs of chromosome that contains human genome there. So genetic information are in the genetic information. So chromosomes are made up of DNA. Chromosomes are made up of DNA, and one piece of DNA is called gene. The instead of the DNA, we will see there's 4 different types of the nucleotide. If you closer look at the strands of the DNA, you will see it made up of all different types of nucleotides, which are presented here by initial letter. Like C stand for cytidine, G for guanosine, T for thymidine, A for adenosine. So this is a background knowledge to see the DNA. DNA has a double-stranded DNA. So that adenine, guanine are the sequence like this. So amplification technique for cloning that specific target parts of the DNA sequence to generate thousands, millions of the copy. So DNA is a stable. DNA has a power, and also we can copy easily. The color of our hair, our eye, or our skin are determined by genetic information stored in DNA. So there are 2 advantages in storing genetic information in DNA. First, DNA is very stable. Secondly, this is easy to copy. So storage here, DNA, double-stranded here. So this is reliable. Okay. So genetic information, we can get it from the sequence of that nucleotides. So that genetic alphabet. So let us see the nucleotides are like words in English alphabet letters we used to write genetic information. So that nucleotides do not have any meaning like an English alphabet. So it's when words only have a meaning. So that begins to make sense when number of letters to put together in proper order or sequence. By putting together in a particular order, so we have words. So that makes sense. To give an analogy, nucleotides are similar to letter, genes which are single continuous strands of the nucleotides, which is sentence, and chromosome, which can be made out of gene, that sentence. So it's like a paragraph, and genome is the made out of the chromosome. It's like a book. So we can follow that all genetic information as we write the whole sequence on a paper. Human genomes has contain 3 billion nucleotides. So that would fill 200 copies of the thousands pages phone books like that. Essentially, DNA from different animals look at the same. So some organisms such as virus have a RNA instead of DNA. So we will get that later. So now we will explain about the DNA first. In DNA, which there's a double-stranded fragment, nucleotides from one fragment form the chemical bonds, its counterpart from the other fragments, so which is called the base pair. in human DNA, 3 billion nucleotides. If we write a whole sequence, so let us study uh, so when we getting the specimen to test the virus, the specimen collections is we can take it through the throat swab like this. So samples collection is usually very easiest way is the throat swab. This one we can extract from that uh, specimen, we can get the DNA. Also, we can get the specimen collection from the blood as well. So now we starting material will be specimen is blood. So when we take it out the blood, so we can do the viral load testing. So sample collection will do like this. So blood of the infected patient containing virus, after taking blood from the patient, we centrifuge for a while to get a plasma so that unwanted cellular components of the blood are separated by the centrifuge. So only plasma or serum we can get it. So when we get the virus in the plasma or serum, the next step is to take it a DNA out. The process of taking DNA out is called the extraction. So how we will extract the uh, what is it? The specimen, we will extract it out the virus, the infected plasma containing virus. So viral load testing go like this. So there are many methods to extract the DNA from the virus. Now we will use the this is called the Quanjing extraction kit. The kit name is it's made from China. So Quanjing extraction kit, which have been approved by relevant regulatory authorities to be used clinical testing. So after we getting plasma containing virus, first we need to chemically destroy it. So there are the steps: lyse, bind, wash, and elute. The 4 different steps called lyse, bind, wash, and elute. The lyse is the first step. We chemically destroy virus by buffer. After that, proteins mixed with the DNA, fragments are destroyed by protein kinase, so the DNA become free of the protein. Then we pull the buffer into the tube and to separate the DNA from other cell fragments. This is a lyse step. After adding the buffer, to optimize binding, the condition between nucleic acid and silicon membrane in the tube. So DNA became free of protein now. So then we pull buffer into the reaction tube and to separate the DNA from the other cell. So we pull the contents of the tube into another tube, and that has a silicon membrane to capture the DNA in order to optimize the binding condition between DNA and the silicon membrane. We pull that buffer prior to the step. So the spring and column on the centrifuge, then unwanted contents of the membrane will be in the rubbish container below. So at that step, we will get a purified DNA in the collection tube. In this extraction step, DNA or RNA in the virus and patient plasma are extracted into the collection tube. So we can also do some virus like hepatitis C virus, HIV, RNA virus, or the other RNA instead of the DNA. And then RNA also made of the nucleotide end, but only one strand. DNA has two strands. Now we have purified nucleic acid. But the amount of viral nucleic acid is so little that we cannot do sensitive testing. So that is why we need to do the amplification. That step is called amplification. Amplification is amplifications of DNA of the organism of the interested. And among many nucleic acid amplification technology, polymerase chain reactions are the most common used method. So we amplify DNA because large amount of DNA is easy to be identified. The earlier presentation, we have learned that 3 billion nucleotides in human genome. So similar amount of nucleotide in viral genome as well. So virus in practically we cannot we realize that copying the whole viral genome is not practical. So we have to find a way to make amplification process more efficient. So each individual has a unique DNA sequence. So each animal species has their own DNA unique. So by looking, so we need to determine the organism. So we increase the number of nucleic acid by means of the amplification. So the if amount of viral nucleic acids is so little, so we can amplify, and practically we need to be identify the what type of virus, any virus. It amplifies the specific fragment of the nucleic acid. So we can identify, looking at the DNA sequence, also we can determine organism. Likewise, every animal species has its own unique. The same principle true for the virus we interest. So each virus, coronavirus, has the unique DNA sequence. By looking at section of the unique nucleotide sequence, we can determine whether it is belongs to which virus, like HIV or what type of virus, hepatitis C, or the amplification segment of nucleic acid. So he is the scientist, inventor of the nucleic acid amplification technology called polymerase chain reaction, PCR in short. This is a simple, rapid, and sensitive and versatile in vitro method to find the sequence of the DNA from an initial complex sources of the nucleic acid. In PCR, there are three different steps: denaturing, annealing, and extension. What we need for PCR is a primer. It can be specified for certain bacteria species and the genes. We can specify. At room temperature, DNA is stable. At starting point of the PCR reaction, segment to be amplified needs to be in double-stranded form. Therefore, single-stranded RNA, we need to build the that we have what we will get there RNA. So once we raise the temperature at 49 degrees Celsius, double-stranded nucleic acid denature. The two strands become separate from each other, as shown here. As mentioned, we do not want to copy the whole genome. So we just want to copy the segment of the DNA or RNA, the virus we are looking for, and we need to know the starting point and the ending point of the unique segment of the nucleic acid of that we interest virus, which is called the primer. So to look for the starting point and the ending point, we will use primer. The primer are single-stranded piece of their DNA, typically 18 to 30 base long, and the specific site is we call template. The template is the technical term that used by molecular biologists to refer that segment. That will get the template. So so then we will extend. So template has a nucleic acid unit of that virus. We need to remember this is the nucleic acid of the whole genome of that coronavirus. As mentioned, we do not want to copy the whole genome. So we just want to copy the segment of DNA, RNA, unique to the virus, what we're looking for. We need to know starting point, ending point. So like this is a template. The primer extension. So one cycle of PCR at the end. So there will include 3 steps: denaturing, annealing, and extension of the template. So we have amplified that one DNA to 2 DNA strands. So each PCR cycle will be goes by amplification cycle by cycle. So each cycle lasts about 1 to 2 minutes only after duplicating at the temperature of 72 degrees Celsius. By repeating PCR cycle, many times we can amplify DNA fragments by hundreds of thousands of copies. For example, if we repeat PCR cycle for 35 times, we will have the 34 billions of the applicons. A technical term used the molecular scientists call the amplified DNA fragments. So template will go first cycle with 2 copies. And then 2 copies squared by 2 is a 4 copies. 4 copies squared by that cycle will be in 8 copies. 8 copy will goes to 16 copies in the 4th cycle. So it will goes to 35 cycle. So it will long last for not more than 40 minutes. So one cycle is 1 to 2 minutes only. So exponential amplification, there will be 34 billion copies. We will get it after logarithmic amplification. 2, 4, 8, and 16, then amplify. Finally, we got the 34 billion at the 35 cycle. In order of PCR amplification to happen, we need to addition to the primer, we call is the Taq polymerase buffer. Taq polymerase is an enzyme that is responsible for amplification. It uses that NTP available reaction chamber, so form the complementary strand to be extended. So at that point, we can have a look diagram to keep track what we have been doing, and we start with the patient blood virus, right? And then spring down the blood cell, taking plasma or serum, and then extract the DNA out from the sample. And now it's time to come for the that virus detection. So if we know how many fragment nucleic acids now we have, and we can calculate how many we had before amplification based on the number of the PCR cycle. So we just run. So to count how many target DNA fragments are what we use. By repeating PCR cycle many times, like 35 times, we will have 34 billions of the applicants. So it's sufficient to amplify about the 25 to 39 cycle. So it only lasts about 1 hour or 60 to 90 minutes only. So it provides the optimum condition with the pipetting step only. So instead of running applicant in a gel, we use the fluorescent the color. So these conventional PCR, we use fluorescent to detect them in real time. In real-time PCR, dye with the fluorescent dye, so that will bind to the applicants, release fluorescent signal when new applicants are made. So amplification show us increasing the fluorescent intensity, so we can calculate the viral load. So we can calculate how many we had before to count how many target DNA fragments, and the amount of product have been measured. And by recording that fluorescent color, so greater the amount of the product, the higher the level of the fluorescent. So which have no viral copies. So we will take those standards to do extraction amplification, and then we do that sample. Greater the amount of the product, higher the level of the fluorescent. So in this graph, we have strength of the fluorescent on the y-axis and cycle on the x-axis. So mathematically, we can plot this fluorescent versus cycle number. Advantages of PCR is quick, reliable, sensitive, easy, and specific. Disadvantages: need for equipment, Taq polymerase is expensive, contamination, forced reaction, capacity, and specific amplification. So restriction of the PCR, contamination of the reagent or lab result in false positive or result failure due to the mistake in the protocol. So not only the coronavirus, we can also detect that method in HIV, H5N1, swine virus, SARS, other bacteria, and analysis of the other genes. This graph is to help us to see the whole picture of the viral load detection using the real-time PCR. So we started off with the plasma or serum of the patient's blood and extracted DNA or RNA from the plasma or serum. We use the PCR method to amplify the segment of the nucleic acid unique to the virus of interest, and we detect the fluorescent signal to determine the that critic value of the sample, and then we plot our sample against a known standard critic value. There we derive the number of virus. Okay, so my presentation summarize is this slide. So thank you.
All right, thank you, Dr. Dali, for your wonderful presentation about how convenient is PCR test. So hope you guys are very clear regarding the presentation presented by Dr. Dali. So if you have any question, you may address it to the comment section and Dr. Dali will answer your questions. Anyone? So we do have one question here from Thomas Alva Edison. Hi, doctor. What is the difference between PCR test and serological test, ELISA, and rapid antibody test?
PCR test is we can clearly define the viral load count, molecular. Serology test is showing only positive, negative. So present or not, present, absence, positive, negative, that's it. Serological test. ELISA test is we can do mass sample. So once PCR test has ran, at least specimen must be more than 200. So 500 of like in the public community, we can do thousands of the specimen. So it's time saving. ELISA, assay, enzyme, link, immunosorbent assay. So it with the biosafety is also different. So PCR test, serological test, and we can do in the community ad hoc. Rapid test is, as name itself is rapid. So only after antibody produced, after the window period, antibody only we can test. So this is rapid test. It's a spot test, something like a pregnancy test, which is called the rapid test. But PCR is the best gold standard. We can also test the antigen. No need to wait for the antibody producing. And then more sensitivity, specificity, reliable in PCR rather than rapid test. Rapid test has so many false positive.
Okay, so hope that answered the question from Thomas Alva Edison. So there's another one from Sabik Ahmed. He mentioned that, shall I know about the new mutation virus from UK?
Okay. Coronavirus has a first wave, second wave, third wave is going on. There's a new mutation virus is occur in the United Kingdom. I also know, but I don't know exactly. This is very updated and very recent, but it's with fungal infection also. Genes with that fungus infection. That is why they are still studying. So as now, so there's a vaccines are approved. So almost 95% are safe. So human mammalians, not only humans, mammalian can be protected by this vaccine. for coronavirus. But new mutated vaccines occur in UK because the virus itself has the antigenic shift and antigenic drift. So maybe next year another mutation like the influenza virus can be happen. So that vaccine should be also update all the time, auto update. We must be very careful.
All right. Hope that answered the question by Sabik Ahmed. So, anyone want to ask some questions? So, if there's no other questions, so I would like to end this webinar by thanking Dr. Dali to be our speaker for this webinar. And thank you, MAHSA University, to give us this opportunity to deliver how convenient is the PCR test. So, thank you, everyone. Please don't forget to follow MAHSA University Facebook page and also Instagram for updates regarding our upcoming events and webinar. If you do interested with our program, please do visit our www.mahsa.edu.my and contact us for your interest program. Thank you. Thank you, everyone. Thank you, Dr. Dani.